In this video, we'll be focusing on the mechanism of phagocytosis. I am sure you are really clear that what defense mechanism we have in our body. As you all know, our body has physical barriers such as skin and a mucosal layer that prevent the entry of bacteria into our body. However, for most of the time, the bacteria or pathogens still manage to enter our body. There are several reasons. Firstly, the pathogen might be resistant to some chemicals produced by our physical barrier, or in most common case, they enter through a wound when we are injured or we have a cut. After the pathogen enters our body, the next line of defense is phagocytosis. Phagocytosis is performed by white blood cells circulating in our blood, tissue macrophages, and neutrophils. Tissue macrophages can also be called monocytes. The distinct difference between neutrophil and monocytes is the shape of the nucleus. As you can see, neutrophil has 3 parts and monocytes have one big nucleus. You can also call them phagocytes. These white blood cells are present in the blood, and some are attracted to the extracellular area by the chemicals released by the pathogen itself. These are called chemokines. But you don't have to include this level of detail in this course. Let's go straight into its mechanism. First, we have a pathogen enters the tissue. The phagocyte is attracted to the pathogen by the chemicals of the pathogen. These phagocytes will move along the concentration gradient, and we call this chemotaxis. The phagocytes itself have several receptors on the surface that allows it to bind to the antigen of the pathogen. Once the phagocytes bind to the pathogen, it starts to change its shapes and engulf the pathogen. The pathogen is enclosed by a space in the phagocytes that we call a phagosome or a phagolysosome. Both terms are correct. At the same time, the lysosome in the phagocyte will migrate towards the phagosome. The lysosome releases their chemicals, lysozymes, into their phagosomes, and this hydrolyzes the pathogen and kills it. The hydrolyzed products, or debris of the pathogen, can either be absorbed by the phagocyte itself or excreted through exocytosis.